I hope all of you have the appropriate stimulants to keep yourself from nodding off in the next hour and a half. We are going to move to our panel on neuroethics and novel treatment neuropsychiatry, which will be moderated by Barbara Sahaki of Cambridge.
So it's very nice to see a lot of young people in the audience, young scientists. So I hope that you'll take an interest in this area and consider doing some neuroethical studies in the area of mental health, neuroscience and mental health disorders. So neuropsychiatric disorders such as dementia, depression, and conduct disorders are debilitating conditions which detract. And there is a very great impact also, including financial costs on society of these disorders. So we really need to find better treatments for these disorders so that we can help people have a good quality of life, a good functional outcome, and also we can reduce the financial burden on society. So neuroscience has been critical to the understanding of the brain and health and disease, and in developing more accurate diagnoses and new treatments across the lifespan. In this panel, we're going to discuss important novel treatments for patients with neuropsychiatric disorders, together with the ethical issues that may arise from such treatments. And we'll also hopefully consider a vision for what holistic treatment might look like in the future, and also changes in the patient-doctor relationship. So I'll introduce the speakers, and then I'll ask them to sit down in the front row, except for the one who's giving the talk, so they can actually see the slides. And our first speaker is Dr. Husseini Manji, and he'll discuss new holistic approaches by pharmaceutical companies such as Johnson & Johnson Janssen towards mental health. And he'll talk about some of the neuroethical issues that may arise with the use of neuroprotective agents for the treatment of Alzheimer's disease, such as early detection and screening of elderly people. Our second speaker, Dr. Helen Mayberg, will discuss the neuroethical issues of chronic debilitating conditions such as depression and the impact of these conditions have on the brain and functional outcome. She'll discuss the possibility of brain stimulation when drugs are no longer successful in the treatment of depression. And she'll consider the new role that medical and device companies may have in terms of providing a comprehensive approach to improving mental well-being and the role of adjunct rehabilitation. And she's also going to talk about how these devices alter and to some extent change the relationship between the patient and the doctor. And our final speaker, Dr. Georges Moll, will discuss novel approaches for non-invasive neuromodulation with a focus on functional MRI neurofeedback. And this emerging technology will potentially allow scientists and clinicians to interfere with brain networks involved in complex psychological states, including empathetic feelings and anger, with possible applications to neuropsychiatric conditions such as conduct disorder, psychopathy, and frontotemporal dementia. And of course, modulating these complex emotional responses in these patient groups raises a possibility of unknown effects. And so we'll be discussing some of these ethical questions. So I want to thank the speakers, and we'll first hear now from Dr. Husseini Manji.
Thank you very much, Barbara, for the introduction, and even more importantly for the invitation to be here today at this very important meeting. I really think we're at an amazingly exciting time. I think there's no question everyone would agree that there's nothing more complex than the brain, and we're far from where we'd like to be in our knowledge and our understanding, but we're making a lot of breakthroughs. But I think what this society is trying to address is, as we make some of these breakthroughs, we're going to have to grapple with some of the ethical questions that we probably didn't even have to until, sort of this day and age where we're coming up with the possibility, as Barbara said, of potentially diagnosing people on their way to Alzheimer's, prodromal schizophrenia, etc. I'd like to talk about some of those issues. Again, I think most of you know this sort of data. This is an editorial from the New York Times by a former Supreme Court Justice and a Nobel laureate. It's kind of taking the point that, while we're in this time of deficit reduction, etc., unless we solve some of these devastating illnesses, we simply won't be able to sustain the cost for some of these diseases. And also, disease. Today we think about 30 million people in the world have the diagnosis, probably at least as many have it but are not diagnosed, and with the aging population the number is expected to triple by the year 2050. And the estimates, and this is more about humanity than cost, but just to give you an idea of the cost, the estimates are that the US is going to cost $1 trillion by 2050 to look after individuals with this disease. Many of you may know some of these kinds of data, again looking at serious psychiatric illnesses, and the World Health Organization has commissioned some of these studies asking the question, what are the diseases, all the diseases that affect humanity that have the biggest disabling impact? And the way they've quantitated disability is by something called disability adjusted life years. So if you sort of die from an illness, you lose those life years, but also if you're ill with these illnesses and not able to function to your capacity, that counts. And you can see that the serious neuropsychiatric diseases in the Western world are by far and away the leading cause of disability. And why would this be? I think partly it's because what everyone knows, these are actually far more common than people sometimes appreciate. Probably the biggest factor is that by and large they hit individuals when they're relatively young and they're lifelong, so they're sometimes viewed as the chronic illnesses of the young. On the right-hand side is just, again, something that was published in Reuters about 3 weeks ago, looking at the cost of taking care of these, cost to society in Europe. The estimate is that today something like 800 billion euros, or $1 trillion US dollars, and it's thought to get just worse and worse. So these are devastating illnesses. And I think most people probably also know these illnesses also have a huge impact on what we consider physical health. So in the US, it's thought that people with serious mental illness might have a lifespan that by up to 20 years compared to someone without those illnesses. So it's something that we really need to take seriously and do something about. This is something that I think Barbara Sahakian has been one of the real pioneers in thinking about. The fact that yes, we need to talk about these devastating diseases, but to me and Barbara and many others, any society's greatest resource is its cognitive and mental capital, our brain power, not oil or minerals, etc. And yet, while we've had this appreciation about trying to do everything we can about physical health, what about mental health? So what about age-related cognitive decline that probably most of us, unfortunately, will undergo? What about learning disabilities in childhood that may prevent people from reaching their full potential? And it's not talking about putting Ritalin in children's lunch boxes, but are there non-pharmacologic ways to help them, even if they're not hospitalized with these diseases or conditions? It may prevent them from achieving their full potential, and that's something we have to grapple with. As I said, I think, and if those of you attending Society for Neuroscience meeting this year, you'll see we're making tremendous breakthroughs in our ability to sort of look at neurons individually, look at the expression profile of different genes in them, look at plasticity, it's sort of the dynamic changes in neural circuitry in living animals. And in humans, we can also, there's a lot of advances in neuroimaging. So there's still a long way to go, but we do believe we're really making advances that we weren't previously able to, and that's starting to translate into potential novel therapeutics. First thing I'd like to talk about is Alzheimer's disease. If you look at the top left-hand side, this is something probably everyone knows, a devastating neurodegenerative disease where over relatively short amount of time, neurons, nerve cells die, and you have this progressive atrophy or shrinkage of the brain. If you look at the bottom, that represents a post-mortem human brain with what's shown there as an amyloid plaque, this thing that looks like a spider is not supposed to be there. And for over a century, we've known that patients, post-mortem brains from patients with Alzheimer's disease have these amyloid plaques. But for a long time, it wasn't known whether this was in some way causative, or is it just a scar? When neurons die, you deposit amyloid. And then a lot of progress in human genetics from John Hardy and others suggested that most of the genes that are associated with early forms of Alzheimer's are involved in amyloid processing. So that led to a lot of interest in the possibility that amyloid might be causally involved. If you look at the third panel at the top there, that's what scientists have done, is they've genetically engineered mice containing some of these human genes. And what they find is that the mice are normal when they're born, but as they grow older, they have this amyloid deposition that's shown in the brown staining, and they show some learning and memory deficits and some long-term potentiation deficits, etc. Clearly, they're not perfect models of Alzheimer's disease, but that suggested that potentially, as you develop or deposit amyloid, it contributes to some of what we see in Alzheimer's, which begs the question, if you could remove it, would you help people? And so on the extreme right, you see what's, as an example of using a monoclonal antibody to remove amyloid from the brain. And you can see in this study, in the mouse, were very successful in removing it. And in these mice studies, you also showed some cognitive benefit. So that's led to human studies. And this slide is a very brief summary, lots of caveats in terms of statistics, the subpopulations, etc. But this is suggesting, that from these phase 2 data, that there's a possibility that by using these amyloid removing agents, you may be in a position to slow down the rate of cognitive decline. And the slide shows the results from an APOE4 non-carrier population, which is thought to be perhaps less further along. Time will tell whether this holds up in phase 3 studies. So there's ongoing studies with antibodies like this, with vaccines. We'll find out. There's a lot of biomarker data. So this is some of the biomarker data on the left-hand side, visualizing amyloid in the brain and showing that over the course of 18 months in patients with Alzheimer's disease, it continues to go up even in patients with what's known as mild to moderate disease, whereas with the antibody treatment, it goes down. And on the right-hand side, phospho-tau is a biomarker you can detect in the spinal fluid, and many people think it might represent a marker of neuronal degeneration. So as neurons die, you see an increase in phospho-tau, and that seems to be going down. So as I said, at this point, it's still preliminary, but hopefully we'll know relatively soon whether these treatments work or not. But almost certainly, even if they work, they're going to work better the earlier you can get in. So, coming in after someone has a diagnosis of mild to moderate Alzheimer's disease, probably this disease process has been cooking in their brains for 15 to 20 years. And it may be that you can only do so much good at that point. So more and more there's an interest in moving earlier. And today, when you have a diagnosis of mild cognitive impairment or prodromal Alzheimer's, the biomarkers aren't actually that bad to sort of have some confidence in predicting who's going to go to Alzheimer's, who's not going to go to Alzheimer's. And the FDA is working on some of these biomarkers, as is its consortium called ADNI, to help us understand, okay, who is on their way, where if you intervened, it might help. But obviously, the ultimate goal may be to identify people long before they even have symptoms. And that's obviously going to create a major ethical consideration. If the treatments are as safe as water, then it may not create much of an ethical dilemma. People who you think are on their way, you could treat. Almost certainly, the treatments are not going to be that benign. You're talking about serious diseases, the treatments are going to be serious. So how are you going to sort of make these decisions as to the risk-benefit ratio? How are you going to sort of get people, people over 50, screened with some of these and ensure that they're not going to get discriminated against? Because, while we'd like to believe we live in progressive societies and employers or others aren't going to discriminate against people who are on their way to one of these diseases, we need to safeguard against that. So I think that's something that until a few years ago we really didn't have to worry about, but it may be reality for us. It may be that we're going to have treatments that can start to slow down Alzheimer's disease, that they're going to be the most effective the earlier we get in, but that opens a whole can of worms in terms of some of the ethical considerations. Many people, and hopefully including Steve Hyman in the front, believe that we're in a very exciting time in terms of psychiatric diseases. And this is just, again, it's just headlines from different journals. But again, sort of trying to make the point that we really are making advances. We've got a hell of a long way to go, but we really do think we're making advances, and some of these are turning into potentially novel treatments. One of the diseases where we really need to do something is schizophrenia, which many people consider sort of kind of one of the worst diseases you could have. It afflicts something like 1% of the population. By and large, hits people when they're very young, usually in their adolescence, etc. And unfortunately, the present treatments we have do, I'd say, a reasonable job if people stay on the treatments of controlling their hallucinations and delusions, but we don't give people their lives back. And that's, what we need to try and do. Well, this isn't definitive. More and more data is suggesting that while schizophrenia isn't a classical degenerative disease like Alzheimer's, when you do a number of the brain imaging studies, or you look at postmortem brain, you seem to see what looks, what is depicted in the cartoon on the left. That's bad news, good news picture. The bad news, the good news, let me start with, is that the neurons, by and large, don't seem to be dead. The bad news is that they seem to be atrophied, so, fewer dendritic spines, less branching, etc. And more we're also recognizing that perhaps a lot of this actually happens relatively early in the illness, and that some of it, may happen as a consequence of repeated episodes over the 20 years, but some of these things happen relatively early. More and more data suggesting that the cognitive deficits in schizophrenia are more related to functional impairment than the hallucinations and delusions, and a reasonable amount of data suggesting that the cognitive deficits may also antedate the first psychotic episodes. So there you're again going to be faced with this dilemma, if we can come up with either pharmacologic or perhaps non-pharmacologic ways to sort of help with some of these cognitive deficits, you might be the most effective before people sort of declare themselves as schizophrenic by having these psychotic episodes. How are we going to handle that? You're talking about, adolescence, you're talking about, if it's drugs, they may have side effects. You're talking about almost certainly you're not going to be 100% correct in diagnosing. So you may be talking about putting some people who don't have the disease on treatments, in order to hopefully help the majority of people. Again, that's a dilemma we're going to have to face. I think some of the work Barbara is doing and others are doing really suggests that there may be sort of ways to intervene non-pharmacologically to help with some of these cognitive deficits. And obviously, many of us think that might be the best case scenario, where early on you want to use things that are the least invasive. And then, later when people do have full-blown illness, almost certainly need medication. But that's something, again, as I said, that we hopefully will have to face because we have treatments that can do something about the cognitive deficits. Depression, you'll hear much more from Helen about her elegant work, on depression. That's, again, something that in, many cases has been underappreciated as to how disabling it may be. And so today we think in the sort of G7 countries, depression is the #1 cause of disability. And although we have a lot of treatments, some data would suggest things like the STAR-D study that only about 1/3 of patients actually go into full remission. So, certainly more patients will respond when we define response as a 50% reduction in their depression scores, but that doesn't mean they're well. And, people who go into full remission, that may be as low as 1/3 of the population. So clearly there's a need to come up with much better treatments. Today the treatments, by and large, the pharmacologic treatments take 6 weeks or more to work. I was talking to Kay Jameson, who some of you may know is a renowned bipolar researcher, and she was telling me that sort of people sometimes describe depression, the pain, as the most severe pain they've ever experienced in their lives. You wouldn't think of telling someone who has bad physical pain, you wait 6 to 12 weeks, trust me, you'll get better. Yet we kind of ask people with severe depression to do that. Obviously that's unacceptable. Suicide is obviously a life-threatening emergency. In the US, we think almost twice as many people die from suicide every year as homicide. There's only 3 forms of cancer that have a higher annual death rate. And yet right now, if someone's suicidal, we basically lock them up, so to speak, to sort of prevent them from doing harm. And yet it may be a time, and the left-hand side of this slide is from Steve Hyman, where we're starting to get, a better understanding that, conditions like depression aren't a chemical soup where you lower a quart of serotonin and you go to the shrink and you get topped up. I think we're learning much more that these are diseases of synapses and circuits, and some of the plasticity pathways regulating the information processing in these circuits may be much more relevant. And so that's led to the thought of, if that's the case, could some of our treatments take 6 to 12 weeks to work because they're starting far from the correct target? Ultimately, increasing intrasynaptic serotonin or norepinephrine translates into changes in AMPA receptor trafficking or NMDA receptor subunit changes, but that's eventually. And if you could have treatments that work directly there, A, could they work faster? And B, could they work better? Because if people have an impairment at one of the steps in this cascade, you can increase serotonin levels all you want. It's not going to translate to the desired outcome. And so there have been studies using an NMDA antagonist, and most of the studies have been using ketamine, intravenous ketamine. And on the left-hand side of the slide, this was a group of refractory depressed patients. On average, they had failed 6 different antidepressant trials, including electroconvulsive therapy. On average, they had been depressed continuously for 3 years. And yet, when they were given intravenous ketamine, starting within hours, there seemed to be a benefit in the depressive symptomatology. At 24 hours, 70% of people met criteria for response. And if you look on the right-hand side, that's generally better than what you see with traditional antidepressants in non-refractory populations at about 12 weeks. Obvious question is this really improvement in depression, or are they just high, etc.? And the data really seems to suggest it's really an improvement in depressive symptomatology. And one question I was asked is, what about suicidality? And probably most of us in this room would think, something like suicide is such a complex psychological phenomenon. Yes, you're in terrible pain, but you're going to be grappling with the consequences in your family, etc., etc. How can you just turn that off? And yet, and again, some of the data Helen's been working on in terms of, the ability to really interfere with certain circuits and have very rapid responses. Again, the data with ketamine suggests about the same thing. Suicidal ideation dropped dramatically, very rapidly. Now, the hope is we'll be able to come up with treatments that, work like this but have no side effects and are easy to take, etc. But I guess one of the sort of pleas I would make is that these are bad diseases. If you want to treat them well, you're probably going to need powerful agents, and almost by definition, powerful agents will come with baggage. I was astonished when I'd be on, university IRBs or even at the NIH, how in some quote-unquote medical conditions, we would accept a risk-benefit ratio that was marginal, and yet for some of the psychiatric diseases, I don't think we've got quite the appreciation about how incapacitating they are. And if you have, too much constipation, that's too, unacceptable. So somehow we're going to have to change the mindset. And again, it's, this idea, if Prozac is going to work for you, you don't go for, the NMDA antagonist. But there's a large percentage of people for whom these treatments won't work for, and these novel treatments may work, but they may come with some side effects. And somehow we've got to grapple with that, given the severity and incapacitation these illnesses cause, that we may have to accept that, and that's something I think that's a sort of shared societal responsibility. As Barbara alluded to, I think more and more we're recognizing, now just last couple of slides, more and more we're recognizing that for most of these illnesses, they're biologic illnesses in large part, and so you're going to need a biologic intervention, but we're talking about people who interact with families and societies. They're not NMDA receptors, they're people. And so the optimal outcome is almost certainly going to involve some sort of holistic solution that includes, sort of psychotherapy, education, work, rehab, etc. This slide shows, some of the things that are being worked on. So I think we're going to be in a position to start to do more about early diagnosis and risk. We may be in a position to sort of monitor adherence to medication, while the person's in the waiting room. We may be in a position to have remote monitoring to start to say, okay, who's on their way to a manic episode based on sleep patterns, based on motoric patterns, based on how much they're on the internet, all those kinds of things. And obviously that starts to bring a kind of a big brother effect into it. On the one hand, I think most people in this room would think, we're trying to do these things to help humanity. That if we can identify people at risk, if we can predict relapses sooner, we could, have better outcomes, etc. And yet, you can see it starts across the line of when are you starting to be a little bit too invasive, and how do you safeguard against this data being used in inappropriate ways, etc. And as I said, before, I think this is one of the things that we think is going to be true for most of our illnesses, that ultimately you want to prevent, but sooner than that, hopefully we'll be able to preempt some of the devastation. But by and large, that's going to require intervening early in the illness, perhaps even without certainty that 100% of the people are going to get the illness. How do we grapple with that cost benefit in terms of helping people, but not sort of subjecting people at risk? So just wanted to give you an overview of some of what I think are exciting advances, but lay out some of the ethical challenges we'll have to deal with. Thank you very much for your attention. Okay.
I'm going to continue this theme briefly. I made slides like 5 minutes ago, so bear with me. I just want to just start by saying the context of the work I'll discuss has both grant support, off-label use of devices, because it's all experimental, with patents that have developed, and some working relationship with a company that is actually doing this work. And we continue to do this work under FDA control and supervision. What I am going to talk about, and the context for the ethical comments, is the development of deep brain stimulation for intractable major depression. And just to point out, the scientific rationale is we developed a circuit model of major depression, identifying nodes in the brain that were really pivotal to mediating the primary depressive symptoms through studies of things as simple as what areas of your brain change when you go from a neutral mood to an intensely sad mood, and then discovering that the pattern of change that goes when you go from being ill to well uses the same networks in the brain. And that led to basically taking advantage of a routine procedure done for intractable and severe Parkinson's disease with the implantation in the brain of an electrode that one can hook up to a pacemaker that's permanently implanted to deliver small and very focal amounts of current to this particular circuit. And basically you can tune this circuit and turn the depression off. And that's done chronically. This isn't like electroconvulsive therapy where someone comes in and you shock them and then they go home and you hope it stays. This is a permanent implantation in the brain, but that showing that patients who show this characteristic pattern with the implantation and the chronic stimulation, you reverse the abnormalities in the brain. Now that's kind of the context for this, but I think that what's important for this discussion is who in their right mind would have that? Well, the people who would attempt to actually allow someone to implant a wire in their brain and wear a battery implanted in their chest and have current going through their brain 24/7 are people who have this kind of symptoms. Hippocrates described it as a fright or despondency that lasts a long time. William James described it as an active anguish, a kind of mental pain that normal people don't know at all. And William Styron described it as a state where psychic energy is so abnormal and so throttled down to nothing that it's like you're immobilized in the state of this supreme discomfort. So when Husseini was talking about the idea of people who are suicidal and what would that be like, just imagine being in this state, which every depressed patient is in, and it doesn't go away. And that's the people we're talking about when you start to wonder about who in their right mind would have a brain implant. And if you need a little more sense of this, you know, every person with major depression, one of the most interesting issues about it, treating it clinically, and I'm not a psychiatrist, I'm a neurologist, but you know, anyone who sees these patients, no patient with a major depression can ever imagine that they will be out of the episode. There's something profound about even a routine major depressive episode where you lose your ability to project yourself into the future. So you can't imagine yourself ever well, and you lose your capacity to actually remember when you were last well. And people have variations in that. And again, William James, should rest in peace, he was so magnificent in his ability to describe his own depression. He described it, I can't, too close. He described it as the idea that the pain is unrelenting, and what actually makes it so hard is that no matter what you do, day in, day out, you can't imagine that you will ever have relief. So these are the kind of patients that we recruited to be the pilot subjects for the study I showed you as the rationale. The patients we had were people that had been in the same depressive episode on average of 5 years. These are people that had failed not one drug, not two drugs, but a minimum of 4 drugs, generally on average, 12 drugs, and had failed conventional and sustained psychotherapy and even electroconvulsive therapy. So this wasn't a capricious approach to treating people. Disabled, off the workforce, off the grid. So this is now beyond the pilot stage. The first 6 subjects, published in 2005, was extended to 20 subjects in Toronto, 3 publications, experience now on up to 7 years, publication on a minimum of 3 years. We get about half the people well at 6 months, and it's sustained. And actually, the longer you're implanted, you just sort of continue to do well over time. At Emory, I have a second cohort with a paper in press, including patients with bipolar II disorder, similar kind of improvement over time. And in fact, in patients who reach remission, they stay in remission, and we don't have spontaneous relapses. And the response grows over time. We have, in our current cohort, 14 out of 17 patients are doing great. And so the issue is company has moved to a pilot study, also the data from St. Jude in press in Journal of Neurosurgery any day, and they've gone on to do a multi-site clinical trial. So we've moved beyond the pilot stage, and that gets to really the ethical issue. This is no longer a relationship between just a research subject and an investigator. The evolution is, and the point of discussion, does it evolve? Where's the point of evolution to where it becomes a doctor-patient relationship? I have to have a scientific equipoise as an investigator. I am both not these patients' doctor, but I am their conduit to maintain a device. And what is the shifting responsibility between a patient and a team, a patient and an investigator, a research subject and an investigator, as you go from a position of having autonomy as a research subject? These people are competent to give informed consent, as we've studied with Paul Applebaum and have 3 papers with Laura Dunn on this topic in this group, but that you actually transition to a varying state of autonomy. And this is where I wasn't going to use slides, so let me just make sure I have this. That this shift is going initially where you're competent but dependent as a research subject because you're so shut down behaviorally, to where as the intervention is introduced and as you are changing over time, you evolve with growing competency and involvement and autonomy. And how do we manage that, think about that? Because what becomes very important is that when you are enrolled after extensive discussions, over a month of interactions with the research team, when you are ill and in this state, all you want is to have the pain go away. Get me out of here. Well, what happens with brain stimulation is there is an immediate shift in the negative angst. It's not an antidepressant effect. The full syndrome is not gone. But something fundamentally changes early, very much like Husseini was talking about with ketamine. There's nuances and differences, but fundamentally there's something in common. That may be all that actually deep brain stimulation does. Gets the car with the parking brake released. And now your job as a subject is to drive. And the brain stimulation allows the car to move, but the investigator or the stimulator does not decide where you're going, what the map is, doesn't fill the gas tank up, and doesn't drive you there. And that's where suddenly the relationship between the team and the subject is altered, and we are now in a new space in terms of investigation of how to best enable that. And so that's what I think we all can discuss, because the issue is whose responsibility lies where. This isn't like a drug trial where if you decide as a patient that you think you're well, I don't want to take the pills anymore, and you just stop. How do you disengage from a stimulator? You have a box, you can turn it off, so you can choose to no longer have the intervention, but now you have hardware in your brain, in your chest, puts you at risk if you needed an MRI scan. Again, it's things can be done safely with supervision, but don't use, don't drive a car without adult supervision. I mean, there are certain responsibilities, and where becomes the point of autonomy of a subject when there are devices involved? It doesn't become that you go on automatic pilot. Batteries need to be replaced. And who is responsible if a patient takes that on themselves and doesn't want to be bothered with being reviewed, seen, doesn't come back for an appointment when they crash, which they will when a battery wears out. So that's the issue, and I'll leave it there.
There we go. First of all, I would like to thank the organizers, especially Steve Hyman, Karen Graham, and Julian Savulescu for inviting me to the board of the society. I hope I can do my best to help in this important mission of this society. And in this presentation, I put together mostly new experimental evidence that I think could be interesting to discuss in terms of neurotechnology and changing our brains in some way. This is pretty new. I can say it's based mostly on previous research from our lab and other labs in moral cognition and affiliation. And so, of course, a lot of things I'm going to show here is still pilot data, but I think it's worth. Well, the first point is that functional neuroimaging, together with the lesion evidence, has allowed us to understand a lot in the last 10 years, especially, to understand a lot more about social cognition and emotion, specifically human emotions. We understand much more about complex cognitive mechanisms, about social moral emotions, and this knowledge, after say 10 years, I think it's ripe to be explored and thought about in terms of how can this knowledge help understand neurobasis or the psychopathology, and also could be employed in healthy people as cognitive enhancement. A couple of words about neuroinformatics, brain decoding. You're probably aware of the several papers which are coming out, especially in the last 5 years. It's a fantastic development which it led imaging analysis away from analyzing single voxels as if they were independent, but take into account the activity of several voxels in the brain at the same time in terms of functional activation, and can also be used to classify and decode, microphone is a bit, to decode anatomical data, to classify patients and to infer cognitive and psychological states. And that's the main point I'll address at the end of this talk. And I'll talk about one particular implementation which is called a support vector machine. There are several others multivariate techniques, but this one has been employed more in with more success recently. This is, I must say that these authors have provided the groundbreaking evidence for using such techniques. Okay, so let's move to the flashy stuff. One of the motivations we had in our research was to understand human motivation in general. And we know that we are moved by several kinds of emotions and motivations such as these, like immediate self-interest. But our species is very interested in very complex types of motivations, which are not, it's difficult to connect to immediate self-interest. We are motivated by a range of social values and situations. Well, in the past 10 years, we've been exploring what is the basis, the neural basis of, let's call it the moral brain. I know it's a tricky expression, but in the past 10 years, we know that we've learned that there is a limited set of brain regions which are consistently involved in moral emotions, moral judgment, and moral behavior from lesion data as well. But the interesting thing is that there are also differences. So if you explore the moral domain in more detail, you can find differences between, for example, different moral emotions. And I'd like to draw your attention specifically to this effect here. When you look at so-called empathic emotions, combining compassion and guilt, we see this basal forebrain effect. And this is something that's coming along over and over now in our last studies and the studies of others. And I think it's a very interesting finding. This is a study we conducted a few years ago at the NIH, in which we showed we had people, normal controls, making decisions in which they could win money or they could donate to causes, to NGOs. And when we look at the common activations, we see the mesolimbic dopaminergic system, VTA, ventral striatum, is highly active, both when you win money and when you donate money, so the warm glow effect. However, when we compare directly donating to pure monetary reward, we see a much more specific, it's a tiny system here, which is, you can recognize here the subcallosal or the subgenual cortex, the same that Helen Meagher was telling about in her talk in depressions. An area very involved in depression, and it's a very interesting link that we still have to understand between affiliation, altruism, and depression. But anyway, we get this very selective activation, subgenual cortex, and in the septal area, bordering the anterior hypothalamus. So, what does it mean? So, I think I jumped one slide. In this study, we looked at, we're searching for evidence, for lesion evidence, for impairment of prosocial or empathic emotions. So in this study, it was conducted in a sample of frontotemporal dementia patients. These patients, they develop early on very severe changes in social behavior. They can be very callous, some of them. And this is the signature of this disorder. If you look here, this yellow effect, this yellow thing is actually the areas which show overall reduced metabolism in PET metabolism, FDG metabolism, as compared to controls. And you see this is well established, the frontotemporal hypometabolism in this patient group. But we're interested to see whether selective or different degrees of impairment would be correlated to specific prosocial emotions. So we designed this task, and it's very similar to the one we saw before, but basically patients read statements like this. Your mom called one night saying that she didn't feel well. You didn't pay attention. The next day she died. Okay, so they are supposed to choose, to pick which emotion they would feel in such situation. So what do we do in the end? We have a range of emotions, and when we look at the basically on the effect of impairment in this task, the selective effect on metabolism. And there what we see, there is what we see. So we see for guilt, embarrassment, and pity or compassion, this is selective involved, associated with hypometabolism in the frontopolar cortex, as well as in the septal region. However, as you see here, embarrassment is not correlated, which is a very nice finding, because these two emotions are the ones, guilt and compassion, which are actually associated with caring for the other in some way, not embarrassment, which is caring about your own reputation. So the story becomes more and more clear as we see that this probably this subgenal and septal anterior hypothalamus region is probably a hub for affiliative feelings. So let me jump now to psychopathy, because of course psychopathy is the exact opposite of altruism. So there have been several studies in psychopathy looking at anatomical changes, looking at metabolism. Psychopathy is underlined by severe and persistent antisocial behavior. A small number, relatively small number from ASPD patients with antisocial personality disorder will fulfill criteria for psychopathy. And the core of psychopathy is characterized by lack of empathy and remorse, manipulation of others, instrumental aggression, and so on. There's no treatment so far for this terrible condition. So this is a study we conducted a few years ago looking at voxel-based morphometry, looking which regions, if there were regions with reduced cortical density, so to say, in psychopaths as compared to controls. And we find anterior prefrontal, this ventral optofrontal cortex, posterior optofrontal cortex, STS reductions. This is reduction in cortex. This is nicely correlated with factor 1, which is factor 1 of the PCL, which is callousness. And this is what we find if you get the same sample and you use machine learning methods to separate the groups. We're able to discriminate with above 80% accuracy for single subjects if they belong to a control group or to a psychopath group. Of course, this is very far for a diagnostic application, but it opens, you can see that it opens several windows into that. So the last part of my talk, I'll talk about a recent development. We've been for the last 2 years developing a tool for machine learning classification and using real-time fMRI. This software piece, we will deliver this as a freely available instrument, probably within the FSL Oxford University package. And this is designed to enable researchers to run these kinds of pattern classifications in clinical as well as in control subjects. It's based on an SVM, supported vector machine, and it'll show the data that we just completed collection. We have the first analysis in which we had subjects engaging into imagination of tenderness, of episodes, autobiographical episodes associated with tender feelings towards family, or in self-pride, hubristic pride. And okay, so subjects were assigned to 2 treatments. So one of them was with feedback. So feedback was given as these circles or these rings. And the distortion of the rings was associated to the projection of the support vector machine. So the more clean, the more perfect the circle was, so the subject knew he was achieving more in this tenderness, the distributed patterns of activation associated with tenderness based on a previous classification. Or the subject could try to attain self-pride. This was cue to the subject. So then, sorry, I have to go back. So these are the preliminary results we are very surprised to see, actually, that although we didn't see an overall increase in accuracy in classification, but we saw a very interesting pattern in that subjects did not receive feedback. They just saw this ring trembling on the screen. They were told that it was not a feedback condition. They just had to focus on the screen and engage on the emotions. So as they got tired, the performance of the classifier is trying to classify each brain volume dynamically. The performance fell down, whereas for tenderness during the feedback condition, it started low and started to rise. And the most interesting finding is that when you look at the brain activations for tenderness in the feedback condition as compared to the non-feedback condition, this is what we get. A very powerful, a very strong activation on the hypothalamus bordering the septal region. So in the neurofeedback verum versus non-neurofeedback. And the last piece of the story is that was really surprising. You use these self-order distinction measure before and after scan. And only in the feedback group, subjects reported that they felt after scan, outside the scan. Of course, this is still very preliminary, but already points to the possibility that people can control the activity within their emotional networks, including the hypothalamus, and they can engage increase empathic feelings. So to conclude, I just want to throw a number of questions and comments. So the pattern recognition and neurofeedback can be used to classify neuropsychiatric disorders, to decode complex subjective states, and real-time neurofeedback can be a unique to decode, to measure deep brain regions, things that cannot be done with EEG. This has been used already for chronic pain and negative states in schizophrenia, ADHD. Some preliminary results are out there. In this, what we're showing here is that self-regulation of complex feelings might be attainable, including with sophisticated and complex moral emotions. And the potentials are in a number of disorders, including depression, which is associated with guilt, and social personality disorder, conduct disorder, drug addiction. It can use possibly increasing empathy in psychopaths. Who knows? It's an open thing. And autism, another condition associated with less empathy. Cognitive enhancement is another potential application, and we all discussed it this morning and yesterday with all the ethical implications, but we have to seriously consider this possibility. You could tune up adaptive motivations and tune down maladaptive motivations, with some risks, of course. Imagine you're treating depressed patients, you could decrease guilt to a degree and so turn into a psychopathic individual. Who knows? So these are just a few questions to steer the debate. And I would like to thank all my colleagues from the laboratory. These are my two kids doing some mental neurofeedback. And this is some pictures of Rio. So those who don't know. Please stop.
So we don't have much time for questions, but we do have about 10 minutes. So do we have questions from the floor?
Thank you for your presentation. And so I'm Paul Ford from Cleveland Clinic, and particularly with the issue of persuasion and obligation for Helen, I noted that you talked about an increasing status of autonomy as people have these stimulators placed. I and I know that you highlighted the question of what happens when they no longer are going to participate in your research. How do you think about your obligation, level of persuasion, risk management for both companies and researchers? I think that you were hinting at some of these things, and I'd be interested to hear what your thoughts are.
Well, I think that, you know, even with the shifting tide with many companies now getting into this, you have some devices that aren't even approved. And then you have routine devices that are made by manufacturers like Medtronic, where the indication may be novel, but the device is available and approved. And that's another layer of complexity I just wanted to throw out there. What I found, and again, you know, we're at the early stages of this, and everyone goes through this. There are many people around, I think even with movement disorder, that, you know, have a device and it's not explanted. Surgery is surgery, and to explant these devices has to be done under general anesthesia. And while it's relatively speaking not a huge deal, something can always go wrong under general anesthesia. So being explanted is not risk-free. There's no data to suggest that having an implant itself chronically hurts you. So having the device in there, you know, when people have died and pathology is reviewed, even with continued stimulation, it's not damage to the brain. So having it stay there. It is a problem if you were to develop cancer or have need for other kinds of interventions to have the device. You can't be put in all kinds of MRI scanners. You can have certain kinds of treatment that involve magnets. So it becomes a complexity for something else. So quite frankly, I've had in Toronto, we had 3 patients that were explanted, 2 because they had complications early on, as described in the Neuron paper, but a third that wasn't better and just felt like I gave it a best shot, I'd like to be explanted. And in Canada, it was easy to explant. When you write these protocols, you don't actually, and when you go to get a grant, you're not allowed to write a grant. No one's going to give you money for the contingency that I'll need an extra 5, whatever it costs, to explant somebody. So, I'm sorry, I'm rambling. Yeah. So
you're not rambling at all, but it's just that there's so much interest. If we could keep the question short.
So let me just keep I'll keep the answer short. The bottom line is that you have an obligation to patients to come up with a way that if they want to be explanted, you help them to explant it. And I think it needs to be an obligation if people are in a research subject and they want that it's taken care of. You can, and patients who then want to have these things, they do take on a responsibility, and you can't stop people, but I think it has to be managed by risk management.
Yeah, just 2 quick questions, I hope, for Professor Mull. I enjoyed your presentation.